Hello.
Hey, Alexis, how are you?
How are you doing, Mike?
Good.
Yeah, so you.
Here, you know?
Yeah, I was on the lecture on WeHi as well.
Awesome.
Where do you live in US?
I'm sorry?
Where are you based in US?
Yeah, Oregon.
Oregon.
Yeah.
Alexis, hi, how you doing?
Hey.
What's with the fuzz? Is this a COVID beard?
What? What is it? What?
What's with your fuzz, Alexis? Is this a COVID beard?
What is fuzz?
Your beard.
Oh yeah. That's a. It's like a back to the wild nerds.
Nice. I think it looks good.
How you going?
I can't.
I'm good. Hey, the
okay,
guys look amazing.
Thank you.
Hey, Mike, you're really soft. I think you might need to turn your mic up.
I think I'm
Do you guys sell the posters?
Not the first person asked me. Maybe we should. We should just give
This is awesome. Yeah, you should give it, man. You should send it with the dice and the reagents, you know?
I don't know if I have them here. Oh, I do have them. We send postcards. Hold on, let me.
Yeah, I can't hear you, Mike.
You can't hear me.
Hey, Alexis, do you want to do the introduction?
No, you do the introduction.
All right, fine.
It will be better.
Postcards.
Oh, nice. That's cool. Who did all this?
It's
all the design.
So I work with a graphic designer. She takes my harebrained ideas and then makes them reality.
That's really cool. Yeah, you can even.
Mike, can you do something about your? Can you do something about your microphone? I still can't hear you very well.
All right, here. Hold on. I think it might be because of the music. Does it work for you now?
No. But anyway.
Really? Okay, hold on.
So how's life in lockdown, Alexis?
Try that. Is that better?
Yeah, that's better.
Okay.
Yeah. I mean, something that I like a lot from the seminar in WeHi, Mike, was the journey that you have been through since you finished the uni, and how you essentially went from a researcher to a CEO. I mean, I think that's something interesting for people here as well. But I mean, I don't know if you had time to actually mention that a little bit briefly, maybe.
Yeah, sure. Can you give me a second so I can pull up a couple slides to speak to them?
Cool.
All right, let's do it. I have no trouble with that. Let me just.
Sure. In the meantime.
Yeah, go ahead, Catherine.
In the meantime, while Mike's pulling together some slides, I might just introduce us. Would that be a good idea? Perfect. Yeah. So, hi everyone from Melbourne. You poor people. My name's Catherine. For anybody who doesn't know me, I have been working in the industry, excuse me, for about 20 years. In Australia, I started off with BD and joined Australian Biosearch and built the BioLegend business basically over the last 15 years in Australia. And then more recently, I was with BioLegend directly for a couple of years and have recently joined Phitonics. I'm really interested in all of the new technologies. Really loved the release of, say, the brilliant violet fluors with Kelly Lunston. I was involved more recently with all of the training sessions around the country for our total seq reagents from BioLegend. And so this new range of fluors from Phitonics is really, really exciting. So I'm really, really pleased to be part of it. So yeah, I wanted to also introduce you to Mike Studnicki. Mike is the CEO of Phitonics, and he was a former vice president and GM for informatics for Becton Dickinson. Prior to that, he was also the CEO of FlowJo. Mike led FlowJo in his transformation to become the leading single-cell informatics company. He also included bringing gene expression analysis platform from idea to scientists in about 9 months time, 9 months in partnership with Illumina.
Wish it was 3 months. That would have been awesome. Wow.
Mike, according to Simon Monard, Mike is a classic overachiever. He plays rugby, competes in track and field, is an ultra marathoner and an avid long distance swimmer. So that's Mike's introduction.
And I am quite literally just finishing up assembling the slides that you asked for, and I'm ready now. Okay. Oh. Cool. Thank you for that too kind introduction. But Catherine's paid, so you have to, you know, maybe Alexis is like, are you sure that's all true? Yeah, so to the point, Alexis, at your encouragement here, I've just dropped in some slides with journey and advice. So hopefully that's useful. Okay, and it worked. You can smell the fresh PowerPoint, can't you? Over Zoom, isn't that how it works? Yeah, so again, Alexis' encouragement here, and I'll make this relevant for people. If you're in academia, if you're out of academia, again, the mindset is the skills are the same. And I don't feel like I stepped away from the bench. I feel like I took all, as we'll discuss, I took all of my frustrations from the bench and started working on them obsessively. And so I'll talk a little, mention that a little bit, and then we'll dive into what's up with our dyes, right? Because it's actually part of this of a thesis that I feel like I'm still working on and probably am obsessed with. But honestly, I mean, when people look at, when they hear a bio or they read LinkedIn, they think, oh, it's magical, like someone had an intent the entire time, and that's 100% not true. I graduated and started a career journey way too late. Way too late. In fact, it was after I graduated that I essentially asked myself this question, which is now what? So with the notion that there really is no finish line, and that just becomes true all the time. And you hear from Catherine being in the industry for 20 years, and like there's just so much more to work on, so much more exciting. Hopefully you hear that from me, right? And the point is to work on things that are excited, maybe narrowly excited as a preview. But I want to share just 8 things I wish someone had told me. And again, I think this applies. It's interesting because we gave a career seminar yesterday morning, US time, and I included some of these, and I had people who were senior in their career to junior. So hopefully some useful things. The first thing I think I want to get out of the way though is a lot of people refer to alternative careers in science. This is some data that's now woefully out of date, but what it is tracking NSF graduates, PhDs over 5 or 6 years post graduation. And in fact, you see the number that in industry now is the largest proportion here. So there's nothing alternative about an alternative career. And I like having that mindset. Now, that being said, okay, now that we've gotten that out of the way, I think the second piece of advice, again, that I wish someone had told me is that get into the business of collecting no's, which I know sounds, I feel like I just said no's, but no's, like people saying that this can't be done. There's a reason that this is important. So my PI told me you should do a postdoc. Business school professor essentially said, no, you need to choose science or business. And pharma consultant told me that I couldn't go in and do essentially what I wanted to do, which is sort of partnering business development and doing that, particularly with a focus on life science tools. And the reason that you've got to realize that these are important is, one, they're highly motivating. I mean, you can tell, I remember these quotes, and it's been more than a decade since I was told these things, but they're also based on an availability heuristic. People only remember what they've experienced, right? So it's not to discredit these people who all had, all were probably well-meaning. But I mean, honestly, I don't remember the encouragement that I got, but I certainly remember these no's. And it turns out to be false. The point is that you really have to begin by writing your own narrative here and saying, but these no's end up being critically important. The other thing too that I always tell people is you also need to start crossing things off your list. So if you're on a career journey and you're looking for a career change or you're looking for career growth, the first thing to do is cross off all the things you don't want to do, right? So start, deliver those no's yourself first, then begin working on those items that you really would like to think about. And how do you do that practically? Particularly in this environment, I'll be honest with you, networking is a thing, but I don't actually like in-person networking that much, but I do love informational interviews, right? So these are quick 20-minute calls with people, right? And I always tell people and I always encourage them, reach out to me on LinkedIn, reach out to Catherine, right? Connect with all of us on LinkedIn and ask for introductions to people who are in roles that you want to be in. right? Same for what it's worth applies for anybody who's coming into your lab who's honestly trying to sell you things, right? Because invariably they know a lot of people across the industry because we sort of get to talk to tons of people. So just maybe a little bit of practical advice. This journey that I had, I worked for Red Bull for 2 years, actually, while I was an undergrad, which was interesting. Actually, was involved in auto sports marketing. I managed a World Heritage site, a UN World Heritage site at the University of Virginia. And then I started working for free. And I think this is important. Your time is cheap when you're in graduate school, and arguably still when you're a postdoc. I always thought I'd be paid to consult. I was wrong. But it's more important that you give that work away for free, because in return are skills that are going to land you your next career. 100%. And this is what I did. I didn't do it with intent, but I knew that I did it knowing that I didn't have all the skills I needed to go and, essentially cross the barrier and leave the lab. I did that for a pancreatic cancer startup. I also got to watch a pharmaceutical company burn to the ground. I started in a 3,000 square foot lab with multiple people. And by the end of the summer there, I was the only employee, which is an interesting experience to see how a business fails. I also moonlighted for a company called NeuroVentures, which was looking at investment, so there's some skin in the game. And then finally, I got formal training. So most of the time in graduate school, like because we're all good at school, we just do more school because we're really good at it. And again, that's a default for us. I encourage people to not do that. I encourage people to get real experience, offer free help, right? And have it be a live fire exercise, and then determine the formal training that you need after you land that job or on the way to that job. It's super important because you're not going to know exactly what's required. I didn't teach myself accounting and finance until far after I was already in business. And it's because I had to. And it was a live fire exercise, right? It's like kind of how you learn when you ran your first flow cytometry experiment, you got to learn quick, right? Like, I think I'll admit it, I threw out my cells when I did one of my first stainings, right? I took the plate and was chucking off the supernatant, and zoom, cells gone. I mean, that was that's a formative experience, so much so that I wrote a Christmas jingle about it called Check the Wells, but I digress. But the point is that's exactly what you need to do here, right? With any of this moonlighting that you do. I think the other thing too is, particularly when I think about this venture capital experience, it was sort of like this movie Training Day, which I know I'm probably aging myself now. But it was intense, and it was definitely something that was very, very different, right? And I asked the venture capitalist who I was working with at the end of the day, I said, well, how do I get into this field? Either being on the pitch side of creating a new business, or being on, the assessing side of being on the investment. And he gave me some advice, which I thought was kind of flippant at that point, but I realized turns out to be incredibly important, which is that nobody cares until you get a deal done, right? And that's true. That's true in any of the partnering pieces, because you can't do it alone, right? Or whether that be a large sale, or until you can put that behind your name in business, nobody really cares, right? And I would say the other thing too that's quite interesting is, even to date, no one really asked me, like, well, and people don't. I get the same thing from my friends in industry. They don't ask you what your thesis was about, right? After you're working for a couple of years or even less, right? They care about what you can do now. They care about your capabilities. The other thing too, and this is an Australia specific slide because I stole this from, borrowed this from a site looking at risk reward, which is something that we're all kind of familiar with. But I wanted to invert this curve a bit and mention that one of the things I tell particularly young graduate students and postdocs is that the more risk that you take on early will lead to exponential reward. This is really honestly a logarithmic plot. If you take low risk on an academic industry postdoc, you will also get very low reward, as you're well aware of. Industry investigator in an established company a little bit more. There's really a gradient around the risk of industry investigator and startup. And this can really go from seed or series A all the way up to sort of just pre-public or post-IPO, right? And there are jobs that are out there, and that or all the way up here, founder. And I think the important piece of this graph is that there are other ways to label this, right? So the first thing I would say to relabel these axes, to put some real meat around this, is that this also scales with equity, which is the largest wealth creator that exists, right? The more you own of a company, the more opportunity that you have. And it does quite literally scale from this. You found it, and you're going to own a significant portion of it, and then on down. The other thing too that I think goes a bit missing sometimes when I talk to people is this is also the amount that you will learn, right? Outside of your own scope, right? And so the more that you need to do other things and want to learn other things that are way outside of your scope, then you choose along that path, right? And I think even that this current business is actually a great example. I mean, the person who maintains our website is me, right? The person who's, spinning up our CRM and maintaining our IT systems. That's me, right? And so that's exciting, right? I like doing those multiple things. I like learning about all those different pieces. So I would encourage you when you think about this. The other thing, though, too, though, that you have to recognize is that you've created a strategy for your life science research. You have done it, right? And you need to realize that you can think strategically. For me, in life science tools, with a bias here, there's a thesis here. And the thesis that I had was, when I was in my short postdoc, was we were trying to cram data together back then. This was before the buzzword of multi-omics. We're just literally trying to smash data together, right? And try to make this work. You realize data analysis and integration is really, really challenging. Joined FlowJo, grew it, sold it. That team is still doing really, really well as part of BD. And then, realize, wow, like the fluorescence infrastructure we have is a complete mess and it's affecting everything that we do. And in many ways, it's not like we've solved informatics, but there's so many tools to leverage there that, there's a little bit less work there. What if we could attack this differently and attack the fluorescence infrastructure and solve for that? But I think the thing that it came to is that the things that frustrate you in the lab, if you want to go into life science tools, can be your greatest source of innovation if you've got the courage to solve them, right? And I'd say too, the other thing to key in often is, what are the things that you think about in your free time? If you're reading about CAR T cells, you're reading about immunotherapy in your free time, it's a pretty good sense that that's what you want to be doing, right? With the rest of your time. You need to act. So this is an active process, not a passive process, and you need to start way earlier than you think you have to. I'm not on career development. I actually think I was probably an underachiever and just pulled it around, honestly, lately. Networking, informational interviews, we talked about that. Also productizing your research, right? I remember being in an interview with a large biotech company and they asked me what I worked on. And I started talking and they cut me off. And they're like, no. You're not talking to us like our grad students. Why should we care, right? And that was kind of shocking to me, right? And so this is how we interview scientists now. I tell them, why should we care, right? Like, I get it. I like research for research sake, but like, why should I care? How's this going to translate? Like, try to productize this for me a little bit. And that's that way I can understand it. Be okay with learning how to be pushy, assertive, particularly in the current environment. I'm gonna be honest with you, people are they're busier than ever before, right? They're dealing with maybe kids at home or too many virtual meetings or what have you. If you don't respond to your emails or your LinkedIn messages, it's probably because they're busy. They're not blowing you off. But honestly, everyone can spare 20 minutes to pay it forward. So just keep following up with them. We're almost there. You need to act because nobody else cares. This is a paradigm thing that I wish someone had told me. The skills you need for building a career, alternative or otherwise, are always the same. Very few of them are taught in grad school, like precious few of them. There's nothing alternative about it. And the difference between who you are and what you want to be is what you do, right? So that's an active process. And maybe the final thing that I'll say here is to mention a high performance team, regardless of what you want to do. If you want to be in academia, if you want to move the needle in any endeavor, you need to either create or join a high performance team. This has a definition. There's actually been a good amount that's written on it. But honestly, no one's Steve Jobs or Elon Musk other than those guys, right? So people read about those guys and they think, oh, I want to be like them. I don't want to be like them. I think they're both jerks, right? No one wants to work with a jerk with a great idea. And in fact, in life science, we've seen this time and time again. High performance team, people have specific roles. The complementary talents and skills, not personalities. I think about this always as like, I want to assemble the Avengers. I want a superhero team, right? I don't want the dream team. I don't want just a team. I want the Avengers. Like, I want people with way different skills and who sometimes aren't going to get along, but are all driving towards a common purpose, right? And in fact, the result of this is pretty amazing, right? And so, this is just some of the results. I call these ticker hitters when we were hitting some of the stock tickers when I was running FlowJo. And we've been on a bit of a tear with Phitonics. We've released, as we'll discuss, more labels this year in the first half of 2020 than have been released in the past 2 decades. And we've done some 40 color flow cytometry. That's not because of me. That's because we have an incredible team, right? Full stop. So, and it's not all drudgery. This is me with a handful of customers that I'm lucky to call customers again, right? Because you get to form just really amazing relationships. So, Alexis, hopefully that was a useful introduction before we go swimming with elephants.
Yeah, it was great. Thank you so much.
You got it. So, and for anybody on there, like, I'm going to put my email up in there. I mean, if you need advice or you want to reach out on LinkedIn, please don't hesitate. I mean, people rarely take me up on it, but I'm like, you got to pay it forward, man. Someone was there for me. So, okay, let's swim with elephants. I feel like we were swimming with sharks. I feel like I should have a swim with sharks section, and like, can't swim with sharks, stay out of the water, and then we can swim with elephants here. So this will be a swimming with elephants. So I think when I talk about elephants, The elephants in the room for fluorescence are this. Conventional dyes only perform in two ways. I'll be honest with you, it was one of our it was our chief technology officer that told me this, and I told him he's full of it, and then he proved it to me, right? And I thought, okay, that's quite interesting. And then he explained essentially, like, why is that so important? And how can then I asked him, well, how can we attack that? The first type of dye performance that we see, and this affects your data analysis, this affects your experimental design, and it affects the fact that when you go onto any machine, any instrument, you're essentially fighting with one hand tied behind your back, right? If you're on a Cytoflex and you're already running, you know, say you have a 16 detector instrument, and now you're all of a sudden you're running 13, a 13 color panel, but it applies all the way up to the high end on an Aurora. You've got 64 detectors and you're starting at a 30 color panel, right? Why is that? Some of it's because of this dye performance type 1. And that is because we have dyes that are broadly cross-excited, right? And you get this incredible amount of spread due to that cross-excitation. And in fact, it limits your ability to leverage that instrument, something that we'll come back to. The other type of conventional dye performance is that dyes can be narrowly excited, but broadly emissive. And again, you end up in the same boat, right? Because you end up having a huge amount of spread, or you end up not being able to leverage all of the detectors off of an array, right? And I think the interesting thing when we consider this, if you will, putting some context around it in the history of flow, is that dyes have always driven hardware development. So if you map out the number of colors we've had in flow cytometry over time, you get the development of the PE tandems, then the development of the 12 color FAX Vantage, the development of the quantum dots, and then in response, the development of the LSR2, the development of the brilliant dyes, and the development of the FAX Symphony. And you would say, well, the world has changed. And I would say, no, it hasn't, right? The development of spectral systems are in response to that. We want to be able to push the plex of our experiments, but we're limited by traditional dye technology. So what do? We use spectral cytometry to try and pull these dye signatures apart. But I think what's really important to sort of mention here is that spectral is not magic, right? And by that I mean, it doesn't remove spillover spreading from our end data analysis. It still makes that a huge problem, right? And so this dye signature, I think is important to say, is the result of non-ideal dye performance, even though you can use it to great effect for phenotyping. But to that point, well, what would an ideal dye look like? An ideal dye would look like this, and you'd be able to leverage all 64 detectors on an Aurora, or all N detectors on any instrument that we have, because it would only emit where you expect it to. It would emit at a brightness that is tuned for your antigen density, because you don't need bright dyes everywhere, right? That would be silly, right? Because you just don't need that. You'd actually rather normalize your brightness to your antigen density and have all of these lined up across all of your different antigens. And if you had a tad less than perfect blue, I'm always tempted to put a trademark here, it would contribute a little bit of spread, but it would still be a very, very clean label. And so that's what we're going to talk about today. So hopefully some useful introductions, you think about it. But let's take a step back. Let's throw that dye performance out. And you know what? Let's throw some additional requirements besides that, because we should be asking for more from our reagents. I almost said, like, can I get an amen? But I realized that'd be a little weird. I was just getting excited. But what if you could design a fluorescent label? And what if you could do it from scratch, right? So I think the thing that we would all pull out, like right off the bat, is we would say, well, it's got to be bright and clean, right? I've set it up a little bit like that. It would have that perfect emission. Okay, yeah. I think it would also be silent in the violet, right? Like we would protect the violet. And we'll talk about that, and we'll talk about what we're doing with violet. But it's funny because when we started talking about this, people said, hey, that's all great. But what about its practical use? And it was very interesting. So we got pushed really hard. Talk to me about lot-to-lot consistency. And I was like, excuse me, right? I want to see this thing be just bang on all the time. But okay. I also want it to be incredibly stable. I don't want to use special buffers for keeping it stable. Okay, interesting. So we'll come back to that too, because I think it's a really important requirement. And the third one here is a new to world capability. And I think it's really interesting. It ties to this thread we've got now developing around antigen density, which is what if you could control the brightness for an individual color, right? And that leads us to really a new place in panel design. How do we do anything that I'll talk about today? So what we do is we take small organic dyes, bind them onto these oligonucleotides, and then we can actually self-assemble them into this Phiton structure. It has this really unique ability where it has these crossovers. This platform is actually what enables us to tune excitation and emission for an individual label, because we can choose the components that we put onto the oligos, and we can change the position, right? And because we're leveraging Fret here, and Fret scales the square of distance, you can actually tune the position for individual component dyes and get the type of clean fluorescence that I'll show you today. But one distinction to make: tandem dyes. Let's just lay these in the ground if we could for a moment. Tandem dyes break down after 72 hours in fixative. They're dead not many hours after that. You know, we leave them in paraformaldehyde, that's it. Tandem dyes, as you well know, right, are painful for compensation. You've got to either use beads, or you just have to sort of pray that you get enough frequency of cells or enough antigen density to compensate them on. These are not tandem dyes. You can compensate them with any marker that you want. We're leveraging FRET, but they are not a tandem dye. The other part that this enables is that I can actually tune the brightness. So I'll show you an example of that, where you can actually quite literally choose your brightness for your underlying antigen. And then it's also really flexible, because what I can do is take a single-strand oligonucleotide hanging off the end of this and then conjugate that onto an antibody. So the way that we've attacked this matrix, if you will, of detectors and excitation is the first 6 labels we did, we said, well, what if you could, instead of leveraging something like a PE conjugate that essentially just messes up everything you've got off the blue and takes some yellow detectors out besides, what if we could go in and stack those detector arrays? And more specifically, why don't we take a look at something like PE Dazzle, which is a canonical PE conjugate, and look at the fact that it is promiscuously excited off the 48, the 532, and the 561 lines, right? And this limits the biology that we can do in our flow. What if we could break that apart and uniquely excite all of our blue labels off of the 48 laser, and then uniquely excite our yellow labels off the yellow laser? And that's exactly what we've done. So we built out a small menu of 3 different fluors off of the blue and 3 off the yellow that are uniquely excited off of those lasers, so that we can go in and stack those laser lines. And it's really fun because we get to talk to customers all the time about, hey, let's take your current panel or your current set of fluors and let's remove, you know, either PE conjugate, and let's go in there and let's actually stack and upgrade your panels. It's really amazing what we've seen there. And conventional cytometry or spectral cytometry is the same. And so, by the way, these are named based on the excitation and then their emission. So something like a 530 is going to be a replacement for FITC. But the other thing, and honestly, the thing that part of the data that convinced me to join Photonics and take this on was what I saw is what I didn't see, I guess, best way to describe it. So what these are here are single stain controls with all 6 of these labels. They're compensated and concatenated. And what you're not seeing here is the trumpet effect that you get when you get spillover spreading. And remember, in this case, this is often a tune, so these are in adjacent channels, and you're not seeing any spillover spreading. I'll quantitate this in a moment, but this, and the fact that it looks like like what you would expect in an ideal system, this is what got me really excited, because I realized if you can do this and do this in multiplex format and do it with very, very clean fluors, the data analysis gets easier, experimental design gets easier. And I'll show you some other interesting things about the physics here when we talk about FOXP3. Also, as a note, I'll quantitate it. But again, we'll get into this fact that we've got 17 unique colors, 19 labels. I did want to show some examples where we pull apart some performance and just look at that for 2 of our dyes. So this is comparing Nova Blue 610 to Brilliant Blue 630. And again, it's not as bright as Brilliant Blue 630, although hold that thought because I could change the brightness of it. But I think what's really interesting about this, when you look at this across all these detectors, is it's incredibly clean, right? There's no emission really in the violet. It's less emissive in the green. So that becomes quite interesting. Yellow, this is a really interesting example on yellow. And it's interesting because we actually have dyes that are even brighter than this now, for instance, out of 700 and 720. But it is brighter than Alexis 694, nearly as bright as PE CF594, which is PE Dazzle, and then brighter than ECD. But again, has this unique characteristic of behaving more like this ideal dye in the notion that it doesn't emit into the violet, it's emitting not as much into the blue, right? And so this is where we started getting really excited. And so again, getting back to this notion of like, let's be quantitative about the impact there. What we did is we looked at spillover and we thought about this in terms of, if you've got one dye that's really, really bad, like let's say, you know, BB630, for instance, has a lot of spill, that's the perpetrator. And all the other dyes you're going to put in your experiment are all victims. What you can do is you can actually sum that up. You can look at the spillover spread in every single channel and say, well, really what I want to see is the victim sum, right? I want to see the sum of all the ways that it's going to mess up the rest of my experiment and my analysis. And so we partnered with NIAID on this and characterized this Nova Yellow dye versus 30 other labels, and it had the lowest spread that they've observed on beads. So we thought, okay, I mean, honestly, we saw this pretty early. We thought, well, maybe we're onto something here, right? This could be really, really interesting because we have the ability to be very bright right? But also to maintain a very low spread and make the analysis easy. And we'll go harder on this in a moment. We try to think about how could you implement this in a really high plex panel and push this to its limit. Similarly, the NovaBlue performed really well in comparison to Brilliant Blue 630. It's not just us that are seeing this for what it's worth. So this is some data from Oliver Burton at the Babraham Institute, just a great customer partner of ours. And I think he was looking at, in this case, replacing PE and some of that promiscuous, essentially, signature that you're seeing here. And his goal in that case was actually replacing these, and because of the cleanliness of any respective version off of either the blue or the yellow-green, right? And I thought that was pretty exciting to see. And we're achieving, we're getting closer to that notion of ideal performance. So to that point, one of the things we wanted to test, there's actually quite a few things we wanted to test. We wanted to test how do these work with other labels, and quite a few of them, right? So in other words, do these work with the Brilliant stain buffers? How's it going to work in that environment? How are they going to work just in combination with a large number of antibodies on the surface of a cell? Let's really pressure test that. Sort of question one. Question two is, can you actually just simply plug and play these labels into an already really pretty packed panel, right? And then, really question three for us was, can you leverage these labels across antigen densities that are high and then also that are variable or low? So that's why we went after some chemokine receptors. And what we attempted to do was plug and play our 6 labels into a backbone 34 color panel and then examine the impact. And again, do that quantitatively. So what we did is, we bought the 34 commercially available antibodies, and characterized, essentially, if you will, the hand we were dealt, right? The baseline panel that we had. And we looked at the spillover spreading matrix there, which is useful for us because, as I'll show you, we're trying to build on top of this and actually, if you will, sort of look at the problem children, if you will, the dyes in here and sort of take them out of the classroom and say, okay, we really want a well-behaved panel here. We'll come back to that. But this is the hand that we're dealt. And what we did on top of that, Seddon in our group ran a really elegant experiment where she took and dropped in each one of the colors that I've shown you out of these 6 BYG labels one at a time, and then said, okay, what's the impact? So getting back to this notion of perpetrator victim, let's drop in each one of these as a perpetrator and see where the victims are and push this to its absolute limit, right? Each one of these is going to be an individual 35 color panel, right? And then we can think about, okay, what's the impact of these? What we see when we put NovaBlue 530 is we get an increase in spread for BB5 and BB515 and FITC, which is intended to replace on a conventional cytometer. So it's exactly what you'd expect, but no other dyes are affected. And so actually on this Aurora, now we're recommending to people just get rid of BB515 because it already has other spread issues. We'll mention that in a moment. But I think the other important part about the slide is, again, it's what you're not seeing. You're not seeing the contribution of spread anywhere else. So you essentially get a free upgrade if you add NovaBlue 610 or 660, because you're not adding spread anywhere else. And you don't see any off-target effects even on the 530. The story is the same for these Nova Yellow dyes, where you see small changes in spillover, right? Where you would intend to replace these in a conventional panel, but you don't see spread addition anywhere else. And so they quite literally become either in a spectral environment plug and playable, or in a conventional environment, you can quite literally swap these out and just go in and stack your detector arrays, right? And we'll talk about how we've expanded that, but we always think about that notion of what if we could leverage every single detector on a cytometer, to the point now where we've got some labels that are only 10 nanometers apart in terms of their emission, which is, to me, is just, it's super exciting to even have those. We shared all of the data from this as well, because I think we're definitely a don't take our word for it type of team. And we took both the raw data as well as the unmixed data and the analysis, and just we made all that publicly available, right? So even if you're gonna go off and do a high plex panel, or you just want to see how these dyes behave, or you just want to have fun doing data analysis, I mean, who am I to judge? You can do that. But I think the other important point that came out for us here was, and we've talked to a number of different folks on this, is now you could leverage these clean floors and have incredible amount of faith in the data that you're getting at the end of this, particularly across, like, for instance, chemokine receptor across all 32 of these severe phenotypes that we looked into, right? Because you're certainly not seeing spread there, right? And it's not the contribution of spread that's getting you there. I thought that was kind of exciting. The work that we're doing now is actually quite literally going in and identifying other problem dyes and then removing them from the panel, and meanwhile upgrading it, right? And so, we're essentially, we're taking out dyes that we think, okay, these are pretty kind of problematic, and then putting some back in. So we went in and just quantified this and said, okay, who are the worst you know, who are the worst in this. Turns out to be BV510 and FITSI mostly actually because of the BV515 contribution, and just not the inability to deconvolute FITSI and BV515. And so what we're doing now is we're taking out 3 of these fluors that are the problem kids in there. And what we're doing, we actually quantify that you can actually remove spread from the panel when you do this. And we remove spread with these. And what we can do is come in and target those antigens with clean fluors that we have in the menu. But we're not stopping there. So this is some work that's in progress now, is we're also adding on to it to create a 45 color panel. And so there's a lot of honestly super exciting work now that we're doing both internally, but then also externally with customers who are now pushing anywhere from 45 to 52 colors. And so we're finally kind of achieving that ideal where we're saying, look, you could leverage 17 colors. And as you'll hear from me quite a bit more, to begin really leveraging that. And it's not just off an Aurora. This is off of any instrument, right? We've been doing the same thing on Fortesses or Cytoflex or on the Tune, right? It's like, let's go in there and leverage all that and do it, if you will, correctly. The other super exciting thing, the fresh data, if you will, is that we had a lot of questions around, because it's a DNA structure, like, hey, how do you get this thing into the cell? Why would that come up? So the size of the structure, it's 150 kilodaltons. It's about 20 nanometers across if you lay it flat, but it actually has some natural curvature. So it's about the same size as an antibody. And so honestly, I was super curious too. Is it going to get in the cell? Like, what's the background going to look like? And so we spent a good amount of time optimizing that, and optimizing a blocking solution to enable that background to get essentially sopped up. Gating on lymphocytes here is RFMO, interferon gamma positive control, so it just shows that we can stain gamma. And then we ran a titration. We didn't go up to saturation on this yet. We're working on that now. But this is interferon gamma, anti-interferon gamma that's bound to Nova Yellow 660, and you see that we get low background and then really good staining on the interferon gamma. This was in a standard fixation protocol. This is, standards like saponin, ICS, intracellular cytokine staining buffers. We also wanted to test, methanol fixation and what could we do in transcription factors. And boys, this is where it got kind of interesting. And so here we're gating on lymphocytes. This is looking at FOXP3 PE, and I'd note, most of the staining here is actually below 10 to the 4. And you're only seeing one population. Okay, so that being said, low background now when we're staining with anti-FOXP3, Nova Yellow 610. Again, we're not up at saturation yet, but what's really interesting is you're both getting a brighter signal, and you're also starting to distinguish both the FOXP3 low and the FOXP3 high populations, which we know exist in there. So we're pushing this a little bit harder now, pushing this up to saturation, actually, and also pushing the event number, of course, across a larger set of samples to show, hey, look, we're actually being able to uncover some of the biology you just miss if you're using something like PE. So it was super, pun intended, stoked to see this. And so some new data that enables us to also leverage these fluors for going inside the cell. And I think it gets exciting when we think about how could you do this and multiplex them and do that off, do that off of channels that might even be near one another. I forgot to mention, sorry, there is a Q&A box down in the Zoom. And if you have questions while I'm going through this, which makes it a lot more fun, please put them in there, and then Catherine will interrupt me, hopefully, and say, hey, wait a minute. So please go ahead and do that. So, and I'll pause after this next part, and we'll just let it go. And I'll certainly save time at the end, so we've got enough time to have a conversation there. But the second part of this, as I mentioned, was, new colors are great. And you can even see me, like, I'm kind of pushing off the other, like the big part of our menu here is, how do they work in the real world, right? Actually thinking, okay, good. How do they work in the real world? And like, what are we talking about in terms of consistency? So we were dealing with, honestly, a lot inconsistency with some of the conjugated antibodies that we had. As you can see, we're building up a lot of different panels. And so we said, look, that has to be a thing of the past. I want to buy an antibody now, and I want it to be the same thing 5 years from now. So we're very public. We publish our manufacturing data. This is actually 2 different manufacturing lots of our fluors. And again, this is one of those examples where what you're not seeing is what's important. And what you're not seeing is any variance across the entire spectrum for these. And in fact, our spec is that if there's 5% variance anywhere in the spectrum, that label just doesn't go anywhere, right? And so we're trying to make that a thing of the past, but we didn't stop there, because this is just on the fluorescent side. And that certainly is a huge contributor to some of the commercially available antibodies that aren't ours out there. The second part of this was, what about did this and then conjugate it to antibodies? So we took all of these, both batches, and then conjugated them to CD4, sent them to NIAID and said, look, we want these to work exactly the same all the time. They've got a really highly consistent instrument. And so we said, look, we really want to be able to compare separation indices across these titrations. And the separation indices, again, looking at these labels on this, looking at two manufacturing batches on the same batch of antibody were exactly the same, right? And no statistically significant differences either off the yellow or the blue. So I'd say we're on our way to achieving lot-to-lot inconsistency, any kind of lot-to-lot inconsistency, at least for what we're putting out, to be a thing of the past. So I think that was pretty exciting. The second part of this was, how hard can you push the platform, right? How does it work in the real world? And we didn't realize this was going to be a COVID relevant experiment, because we ran it just before year end. But what we did is we took an anti-CD3 conjugate that we made, stained fresh cells. That's not a lot of magic. But what we did is we actually we stained and then we fixed cells in 2% paraformaldehyde and left it in that solution for 2 weeks. We didn't fix it and then come back with PBS. We just left it in the fixative, right? So it's a pretty abusive solution. If you do that to a tandem, you will see no signal after 2 to 3 days, right? And that's being generous to tandems. On our side, there's actually no difference between fresh versus 2 weeks in fixative. We've tested this for paraformaldehyde. We've tested this for paraffin as well. And as I mentioned earlier, we've looked at methanol fixation and shown that the labels are also compatible with that. Again, pushing this a little bit harder was, okay, what about our conjugated antibodies? Just how stable are these? So we quite literally left them in a lab bench, light protected, for 2 weeks, and there's no difference from those that are stored in the fridge. So good news for things that we're shipping, certainly, but also just, certainly an indication that because we have this very rigid DNA structure that's holding our dyes in place, right? And we've got our own conjugation chemistry here, that the conjugates are incredibly stable. Catherine, are there some questions? You want to pause here?
Let me just unmute myself. You've already answered them.
Oh, all right.
The question, Eva had a question about ICS, but you answered it. And Alexis was asking about stability at with PFA. And again, you just answered it.
Yeah.
So we could probably address right now the stability at with other fixatives, though.
Yeah, what's been tested? So paraffin, paraformaldehyde, the methanol fixation, the ICS that we talked about, which is fixed perm. And we have not tested other exotic type fixatives, anything other than that set. I would say that we've also tested the fluors in the context of multiple different types of sort of serum and 2,4-G2, FC block solutions, and that there's no incompatibility there. I would also say there's an important point here, which is that there is no special buffer required. Like, for instance, when you're either storing them or making master mixes to prevent aggregation, the same way that some of the polymer dyes work. They just don't aggregate. So we've left them for months, and not over 90% of it is of the mass of these things is DNA. So they're actually quite hydrophobic. So you'd have to try pretty hard to make them aggregate. So hopefully address that question, and yeah, please keep firing away.
Sorry, there's more. My dog's barking in the background, so you're going to have to help me out. How do these perform in IFAs and multiple rounds of laser exposure? As in photostability, I think they're asking.
Yeah, so it's a great question. So we've been working with a partner on immunofluorescence. The fluors, so the data we've got, the fluors are incredibly bright in that application. What we were dealing with, though, because we're leveraging this DNA platform, we did it with not a lot of blocking. And essentially we lit up the nucleus like nothing I've ever seen before, because it was binding on histones. So what we've done now, vis-a-vis those intracellular experiments, is we have a blocking solution. It actually blocks up some of the stickiness of these labels on monocytes, but then we updose that block for leveraging an intracellular. And now we're testing, okay, can we updose that block and prevent that kind of background staining? So I'd say very early days for us in the immunofluorescence world, and it's not something that I'm certainly not an expert in it. So I'm excited to see what we can do. They're testing both frozen and then FFPE. So hopefully that addresses that question. There was a second question in there, Catherine, and I just forgot.
Are we planning to expand into other laser excitation and emission wavelengths?
100%. Sorry, I jumped on it.
That is yes, and we will address that a little bit later. And do these dyes bind DNA dyes?
Yes. So if you're using, yes, absolutely. So they're double-stranded DNA. So if what we recommend is using something like an amine reactive viability dye, because you will absolutely, the dyes have double-stranded DNA. If you're working with some sort of intercalating DNA viability dye, they will definitely stain.
Cool. What about nucleases?
Nucleases, yeah. So it's interesting because of the crossover structure of the actual Phiton structure, the labels themselves are not nuclease sensitive. Now, that being said, the connection that we have to the antibody is DNA sensitive. So two different things that are wrapped up in that now. There are a number of people who have worked with our labels when they do like a collagenase DNase treatment of the tissue, and they just simply wash out the DNase, because at low DNase concentration, you're just not going to see a degradation of performance. So people have done that, and it's not really an issue as long as it's washed out. The second thing, though, that it opens up is you can use DNase now to perform sequential staining. So. Okay.
That's it for now. We'll have time at the end for more questions, I think.
Definitely. Yeah. And this laser question I will hit in this next section and talk to you around some of the work that we're doing and asking you honestly for open feedback as well for what we should target next, because we've been at a pace where we can do about 2 to 4 new labels, at least at R&D scale per month, which is pretty fun. To that end, part of that launch actually was us moving into an arena where we could actually change the brightness of an individual label. So the way we thought about this is like wattage of bulbs, because we had to come up with a new naming convention for fluorophores, and these are actually named after their brightness. In other words, their separation index on CD4. And so we have a label that has the exact same excitation and emission criteria, but you can actually choose the brightness of a label. So we've done that for NovaBlue 610, again, where we've stoichiometrically increased the brightness of that label. And then the same thing for 660. And in fact, what we have now that's essentially in stock and we put on antibodies is this 40S, but now you can go actually 3 times as bright off of the exact same label. That was actually part of a release. This will answer your question, and then I'll get into the laser lines as well, of 13 new labels that we added onto this, right? So it's actually 19 labels that we've done now across the blue, the yellow, green, and the red lasers, and it's 17 unique colors, because actually with 2 of these, we essentially have 2 different versions. We've got the low and the high brightness versions. What does that look like, and sort of how do we work with people on that? With these, the first set of 6 dyes that I described, those we now have CD4 kits for them, as well as conjugation kits, 1 by 100 microgram conjugation kits that I'll speak to in a moment, as well as a set of conjugated antibodies. And I'll show you guys some new work that we've actually done on those, where you have the ability to essentially print any antibody across any color that we have. And then what we're doing is we're actually moving the dyes that we have now. So this is the second set of fluorophores that we've done, really spanning, again, in the blue, kind of going back into the 500s, Nova Yellow, it's certainly moving that Stoke shift out. And we've started in the red really spanning, if you will, APC to APC Psi 5. We're working on further out, right? Like APC Psi 7 replacements, et cetera. It's something that's definitely, it's certainly of interest to a number of folks we've talked with. So that being said, when you have that many fluorophores, we sort of had to come up with a new way of like thinking about them and navigating them. And it's also a question that I get, which is how do you benchmark these? When we do a launch, we're doing this actually during development, but we benchmark them versus current fluorophores, and we then share the data both on spillover spreading and separation index. So we take a look at all of these on an Aurora, and we also look at the plots. Turns out to be a great instrument too for doing just like dye characterization. And then we've actually shared this up on our website. So we have a fluor selection guide up on our website, and it takes a look at all of our labels, all the other labels that are out there, on looking at it across these laser lines. And I realized too, I was sort of like looking at every fluor selection guide, and there were all these really boring PDFs. And so what I did is I created an interactive fluor guide where you can look at individual fluors here, and you can actually select out laser lines, and you can see how essentially all the labels we have compare to everything else that you may be using already, or things, alternatives that you're looking at. And so this was kind of fun, and I also think really useful because, honestly, like why does data analysis get to have all the fun? Shouldn't experimental design also be somewhat interesting. So to that end, we've also moved into, we've got 12 different clones now that we offer across mouse and human. For any of these, we have recommended colors that we have up on our website. But I think the fun thing, and this is like a brand new slide, is we actually have the ability now to print these across any color that we have in stock, which, as you can imagine, now is 6 colors, and then end of this year will be essentially 19 different fluorophores, and then it expands out from there. And so if you order, something that we have in stock or something you're interested in, right, and you want it on a new color, you get it in your hands in less than 2 weeks. And I think that this is just honestly really cool, because instead of it being like a customs program, it's essentially made to order, right? And you can literally just print these on, high yield, it's purified and in the vial. So we've also done conjugation kits. These, I was honestly pretty reticent about conjugation kits, but honestly, we've had a lot of great customer conversations about this. Easy to use, 5 steps, about 24-hour turnaround time. We actually just talking to a customer who just said, look, like it's actually simpler than you make it seem, which is something that you want to hear for sure. And a lot of this too was in response to people just saying, look, like I want to get labels, I want to put them on antibodies now, test them in panels, and then go for it. And so we've had a lot of great use there. So again, CD4 test kits actually for all of our labels. Conjugation kits, similarly, we've offered those as essentially like a 6-pack of some of the first 6 BYG, but we've also broken those out into these 1 by 100 microgram kits. And then we've got quite a bit of capacity, as you've seen, sort of in this ability to print, also on the custom conjugation side. So bringing this all together to answer that person's question around where are we and where are we going, this is where we are now, right? Where we've got these 19 fluors, 2 different versions of the 610 and 660. That being said, I'd say were areas of focus for us. I mean, you can see, obviously, we've got some work to do in the red, but one of the things that we are very actively working on, and when we do land in a laser line, we do land with it's going to be it's kind of similar to what you're seeing in the red, 4 floors at one time, right? It's not just like 1 or 2 floors. So we are working on Purple Rain right now, very actively off the UV and the violet. What I will say here, and I say this every time, please, this webinar, email, what have you, please give us your feedback. It's incredibly valuable about some of the things you're dealing with the violet and UV dyes if you're using those now. Complaints, the good, the bad, and the ugly, we want to see all of it, right? And so we've had great customer interactions there, just saying, these are some of the things I like about these labels, and these are some of the things that I don't. So we're Phitonics. We released, quite a few labels this year. This is the team that puts it all together. I put my email up here too. Catherine's email is very similar. It's catherine.friend@phitonics.com. And then you can also reach us with info@phitonics.com.
Thanks, Mike. So you can reach me. I'm based in Sydney, so we can actually talk with each other as well. Mike, while we're talking, I love this photo. It's so cool. I'm actually going to put it up on the website. I've decided that we have to have a competition to find our aliases for the names of these people. I'm like, Flow Guy, Super Spread Girl, something like that. Although that doesn't sound too good.
Captain Compensation.
Captain.
I feel like that's better. The Sultan of Spread. Anyway, sorry, I'm digressing again.
But Alexis has another question. He was talking about if we've thought about our own line of comp beads.
Yes, yeah, it's such a good question. And in fact, I'll take. So the answer is yes, and it's not in scope for us this year. But the reason that it becomes really interesting is something that I haven't talked about, which is that on these antibodies that we're making with our own chemistry, and also on the conjugation kits, it's a 1-to-1 labeling. And so it's a 1 Phiton to 1 antibody. So it's actually interesting because it's not just compensation that's kind of solved when you have these beads. You could also do quantitative flow cytometry because you have 1 Phiton with a known set of dyes on 1 antibody. And I think that's where it gets really interesting. So we're doing, I'd say, a good bit of work to really run that to ground and develop a strong data packet around that 1-to-1 labeling. And the goal would then be to follow that up with beads to say, hey, look, you can move well beyond this sort of relative expression world, run clean floors, and for all the ones that you have, you can run it quantitatively. So it's something that I get really excited about. I wish we could pull that in. Right now, the focus has just been on violet and UV.
Yeah, can I make a comment?
Please.
I'm hearing myself. Sorry, wait. Let me just take my headphones off. Okay. So something that I found really excited about your your dyes is precisely what you mentioned, that some people might have, I mean, that I'm finding this a lot with our users. So as you design a panel and you go through all the work of thinking of the markers and finding your dyes for your markers, and then thinking of the compensation on the spillover and the spreading that you will get, it happens quite oftenly that once you have the panel design, you want to add something extra. And with dyes that we have, it's actually quite difficult to add something extra. Because suddenly the marker became relevant and you want to add it to the panel. Well, your markers are perfectly suitable for this later addition into the panel that was already thought. The other thing that I like about the markers and the fluorochromes, and the fact that they spill and spread really low, very little into pre-existing markers, is that most color panel, when you actually look at the panel, is what you call a broad panel. So you are targeting different populations that are known, that have markers that are known co-express, and that helps a lot to essentially manage the spreading that you can get. But I wonder how many colors you can fit into the deep panel. And by deep panel, I mean someone interested exclusively on T cells. They don't want to see any monocytes or the dendritic cells. And we have some of our researchers actually interested in deep panels. And I guess your markers, your dyes will be perfectly suitable for that as well. So not only you are not spreading into a broad panel like you have shown in your case, in the example that you've shown, but you can probably make the deeper panel deeper, because you can add extra markers targeting the same population. And even when there is co-expression, there will not be spreading between them.
Exactly right.
So have you tried any kind of deep panel? Have you worked with researchers that are actually developing these kind of deep panels for specific subpopulations?
Yeah.
And how far have you gone?
Yeah, we just got off a call where people are pushing really well beyond 40, either on Ts or I think it was MDSCs, and exactly the way you're describing it. Actually, in two different ways. One was, it was already a deep phenotyping panel pushed to sort of the maximum there. I should say 30 color pushed to the maximum there, again, because they're accommodating for the fact that they're all co-expressed. So you really can't leverage all the detectors. So they were adding in markers to go deeper in an extant panel. We haven't done that on that 45 color on some of the work that we're doing now. Now, that being said, it's interesting because like all the people who are really pushing well beyond 40 now, it is some of that deeper phenotyping. I'd say from like a technology perspective, when we're designing the labels, we'll throw them out if they can't be used in that use case, because we're designing for the entire floor, like we're designing for the entire spectrum, right? So, you know, we've protected the violet, for instance, on purpose so that we could do exactly what you described, because there's a lot more, frankly, there's a lot more spectrum to play with, right? You can also see too, there's a lot more far red we could play with. And I think too, you know, to your other point, to the you know, developing that too, you can do that also. You don't have to buy, you don't have to have an Aurora Symphony to be able to do that. You could do that and leverage the detector array on a current instrument and do deep phenotyping. Because in fact, on some arrays, when you look at some of the emissions here, you might have several options, right? You could say, well, it's 10 nanometer different. You could say, well, actually, for this panel, I really want to use a 700, not a 690. I mean, that's a lot of optionality, you know, where you could space it for a deep phenotyping panel and have that tight control. So anyway, I really like your point, and it's interesting feedback too when we think about, I think particularly post 45, I'd love to be able to see that and let's do a deep phenotyping panel 4850 with the next round with purple labels as well.
Yeah. And the other thing I like about your approach, if I can say something else, is like, when you do panel design, there is something that is already known, which is some of the areas in the fluorescent spectrum are very quiet. Like the FITC and the brilliant blue 515 is an area that will not become essentially a producer of spreading, and it also do not receive spreading quite a lot. So you tend to reserve this to the global markers like CD4 and CD3 and things like that. So if you want to add dyes in the previously empty region between the FITC and the PerCP kind of region, as you guys are doing, you need to do it and also be able to control that spreading, because you still want to have some quiet regions. So that's another thing that I like about your approach, guys, is that you are adding dyes to the spectrum, but you are doing it in a way that you will not affect by producing even further spreading. And that's great that you can control that.
Yes. Yep. Absolutely right.
Yeah, well, that's what we're doing. There's another question from Ricardo. Could these labels with different brightness be suitable for barcoding samples?
Yes, yes, I'm laughing because this is something that we thought we might. This is something we thought we'd be able to do, for instance, like off CD45, and pull that experiment off last year. We were never able to do it, but the answer is yes, because I mean, as you saw based on the data, it's actually pretty. I mean, it's super easy to separate these. So if you've got them on something that has a sufficient enough antigen density for the dim one, I think particularly off that 660, that would be quite easy to separate, right? So I think not only does that become interesting, I'd say too, the other thing as we build this out is we have a lot more dynamic range than I'm showing you here. And so we've been testing some even brighter versions of these to see whether or not you could have a dynamic range that's over maybe even 2 to 3 decades of brightness on a floor. I'll be very honest though, I mean, barcoding is a great example of like, okay, that's a clear fit for that. We're sort of up against the, well, how many different dyes versions do you make, right? And use. And I think we don't know yet. I think where we'll probably end up with in panel design is the point I was making earlier, where we'll normalize our underlying antigen density to brightnesses, right? And sort of optimize that performance. But we don't have any panel design tools, and we've got to work on one where you can control that brightness. Barcoding is, I think, another example of that, where it's like, what's the delta that you need on brightness on a specific instrument to then be able to separate those populations? I'd argue that certainly off the 660, you'd be able to do it. I wouldn't be surprised if you could do that off the 610, depending on some of the blue laser power and what your detector array is. But sure, you could do that now and at least do what? You could do at least 4. So.
Is there any more questions?
Have you guys sent this dyes to Cytec? Because something quite useful when you're designing panels for aurora is that you go to the similarity index matrix that they have, and they own the equation. So I cannot actually do it myself, even if I wanted to get this similarity index to get pre-existing dyes.
Yep. So they had our 6 BYG labels up there. I haven't checked lately whether or not they're still up there. So they had that in there so you could do the similarity index calculations. We've actually been looking at doing our own calculations too for how you would essentially output a similarity index and a complexity index. So I'd say on that front, it's really on us to develop up a proper panel design tool that lines up labels, ours and others, to be honest with you, with the Aurora, and really, if you will, opens that up a little more, to your point, Alexis, around like, well, what's the underlying math here? Because I think the other piece, and it's interesting because this is a cause of a lot of discussions today, is even if you have those metrics, it doesn't tell you much about what you're going to do on the other side when you've got different antigen density and you've got to deconvolute, right? So
yeah,
that's,
I mean, it's one of the tools that I use. Sure,
yeah, absolutely. So yeah, and in fact, we did on the 40 color panel, we ran a lot of similarity indices as we were doing that and trying to figure out the new panel. So it's still, without a doubt, a useful tool.
Yeah,
definitely.
Okay, on that note, if there's no other questions, we'll probably close for now. Alexis, would you like a copy to send out to anyone who can't?
It would be great. That was my next question, actually, because I would like to post a video. I'm sure there were many people that were interested in this and they couldn't make it.
Yep, that's fine. We're happy to give you a copy of the video. And if there's any other questions, please get in touch with me. I'll probably send you all an email just introducing myself, so you've got my email details as well, somebody local for you. And yeah, let me know by return email if there's any questions, or if indeed you'd like to try some, either on CD4 as a conjugation kit.
Awesome.
Thanks for having us, Alexis. I appreciate it.
Thank you. No, my pleasure. We keep in touch.
Thank you.
Cheers. Thank you, Mike.
Thank you, Alexis. See ya.
Take care, mate.